Hello, and welcome to our Lunch and Learn session. Today, we are happy to welcome Casimir to be delivering the Lunch and Learn. Casimir is led by two extraordinary parents who I've known personally for, I think, 16 years, Christine. It's led by Christine McSherry and Mindy Leffler. They've developed a novel tool for capturing disease progression through video assessments from home, which they're going to talk about. We are grateful to support Casimir over the years and thrilled to have them here today. And Christine's also joined by a real superstar panel of speakers. We're going to have an educational component in this Lunch and Learn, and then also talk about the DVA. So with that, Christine, I'd love to hand it over to you.
Thanks, Ryan, and thanks, Pat, for having us back again this year. And you're right, I am joined by some superstars who I've known for a very long time, and I'm thrilled to have them present with us today. Again, we were here last year with PPMD, and you gave us a wonderful opportunity to present the DVA, and so we're excited to present it again. And my screen is always hesitant, as we did in rehearsal, to move. So just give me a minute here. Well, I'll tell you about who's going to be here with us today. So we have Dr. Brian Sen, who is currently at Vertex, and he is a neurologist nerd, as he's self-proclaims himself to be. He's been in the Duchenne space for a very long time, about 20 years, I would say. Originally met him out of Colorado, came to Boston and opened up a clinic at MGH, and since then has gone over to Novartis, worked in SMA and did some miraculous things there, and then came over to Duchenne via Vertex. Sharing that kind of role with him at Vertex is Sasha, Alexandra Cornell. She is a pediatric pulmonologist by trade, and has a lot of pediatric experience, and obviously history in pulmonology, which is so important to our guys with Duchenne. And then lastly, we have Ash Dugar, who I have known for about 4 or 5, maybe 6 years now. Came into the Duchenne space after having some additional rare disease experience at other companies. Originally met Ash at Sarepta. And then he has recently joined Dyne as, I believe, the Senior Vice President of Global Medical Affairs. But we're going to cover a variety of topics today. Ash is going to cover the phases of clinical trials, some accelerated and confirmatory information for us, including some discussion about surrogate markers and where they are, or how we use them. FDA overview is going to be by Brian and Sasha. They'll also talk about endpoints in natural history for Duchenne. And then I'm going to break into the Duchenne video assessment, which is something that we coin as the DVA. So if you hear us to refer to the DVA, it's that Duchenne video assessment. We're going to talk about some of the pros and cons, my experience with it, some experience that I've had with some of our sponsors and some of our source material studies. And then lastly, we're going to talk about an exciting study that will be launched this summer called the ARISE study, and how the community, both sponsors, academics, and our Duchenne caregiver community and participants can help us with that study. And then hopefully we'll have a little bit of time left for some Q&A. Next slide. And again, we already did the introduction, so you know who we are. And I'm going to pass this over to Ash, who's going to start talking about phases of clinical trials. Thank you, Ash.
Great. Thank you, Christine. And hopefully everybody can hear me okay. So I won't have any difficulty with slides because I have no slides for the audience. So I just really want to try and have a virtual conversation and take any questions that come in. So first of all, thank you to PPMD for providing the forum, this forum for discussion and the opportunity to talk about and learn about topics that are so critical for so many people, patients, families, and physicians, and others. I learn a great deal from these sessions and this wonderful conference that PPMD has organized. And then it's always just wonderful to see our friends at PPMD as well. And thank you, Christine. I'm really pleased and honored to have been invited to participate in the panel discussion with experts in the field of pediatric rare diseases, and of course, in Duchenne muscular dystrophy. I continue to learn from all of you, as I have been for many years now. So as Christine mentioned, I'll be covering a few topics, including the role of the FDA at a very high level, an overview of the drug development process, and talk a little bit about the approval pathway. and how it's applied to Duchenne. So, at the highest level, the FDA is responsible for, amongst other things, protecting the public health. And that's really first and foremost. It's really about protecting the public health by assuring that most foods are safe and properly labeled, ensuring that human drugs and vaccines and other biological products and medical devices intended for human use are safe and effective. And I can't overemphasize that aspect of looking out for the public health first and foremost. Importantly, the FDA has a very critical role in advancing the public health by helping to speed product innovations. And it's this point that I want to touch on a little bit later in the talk, and just the tremendous amount of work that they have put in to be able to move drugs forward more quickly over the years. I'm just going to take a little step back into some historical context. So how did the Federal Food and Drug and Cosmetic Act come about? Well, the first comprehensive federal consumer protection law was the 1906 Food and Drugs Act. And that really prohibited misbranded and adulterated food and drugs to be available in the public sphere. And although it was very impactful, the act nevertheless had some shortcomings. It left a lot of products untouched, and there were dangerous items that remained on the market legally. And then in the 1930s, there was sort of a shift in, the political winds shifted, and it was really spurred on by a growing national movement and outrage over some tragic examples of products that did lead to morbidity and mortality. And there was a tipping point in the mid-30s, around 1937, when an untested pharmaceutical agent damaged the lives of many patients, including many children, as soon as it went on the market. So the enactment of the 1938 Food, Drug, and Cosmetic Act really tightened the controls over drugs and food, included new consumer protections for drugs and cosmetics and medical devices, and it enhanced the government's ability to enforce the law, which was quite important. And so this law, as amended, is still in force today. Many of the things that the FDA has put in place have led to sort of the current thinking around drug development and guidance for both non-clinical development, including animal testing, and then clinical studies to properly evaluate drug candidates. And I'll focus on clinical trials here, but I just want to make a short mention that there is a tremendous amount of work that occurs within what we may call in vitro settings, so sort of in cells, as well as in animal models, to really characterize the drug candidate prior to it going into clinical trials. And these steps are extremely important before clinical trials can start. So the term clinical trials or clinical research refers to studies that are conducted in people. And researchers design these trials to answer specific questions related to a drug candidate. And these trials help us learn more about ways to prevent or treat medical conditions. This research has to show that a study medicine is safe and effective before it can be approved for use. And without these clinical research studies and the volunteers who take part in them, modern medicines would simply not exist. So the process of learning and developing an investigational medicine is generally divided into 4 phases. And you'll hear me say sort of traditionally, in 4 phases, because I think that's really changing, especially in rare diseases such as Duchenne. Broadly speaking, at first, very few people receive the medicine being studied. The number of people participating in clinical studies grows along with our understanding of the investigational medicine, and the research continues as long as the potential benefits outweigh the risks. So again, traditionally, clinical trials follow a very rigorous series from early small scale phase 1 studies to larger phase 2, and then to late stage large scale phase 3 studies and then finally to phase 4 studies. And I'll discuss these stages briefly, but a treatment should be successful in one phase, for it to move on to the next phase. And importantly, a successful clinical trial process continues until the developer files a marketing application with the FDA or a regulatory body in another country for the medication to be approved for doctors to prescribe to patients. And it's important to note that the phases and design of clinical trials can be different for certain diseases and specialized medicines, and certainly ones that are often used in rare diseases. So it's not one size fits all, but I'll start with a general overview of each phase for most medications. During phase 1, researchers generally test a new drug candidate in a small number of healthy volunteers, or in some cases, patients in that phase 1 study. And the primary purpose of a phase 1 study is to evaluate the safety of a new drug candidate before it proceeds further. And in addition to safety, researchers can answer other questions related to how much drug is measured in the blood after administration, how the drug works in the body, and side effects associated with increasing dosage in that phase 1 study. In phase 2 studies, researchers administer the drug to a larger group of patients, sometimes up to a few hundred, with the disease or condition for which the drug is being developed to initially assess its effectiveness and to further study its safety. And a key focus of phase 2 studies is determining the optimal dose or doses of a drug candidate in order to determine how best to administer the drug to maximize the benefits and minimize the risks. In phase 3, for diseases affecting many patients, phase 3 studies typically can involve from a few hundred to a few thousand patients for which the medicine is eventually going to be intended for use. So participants are assigned to receive either the medication being evaluated or a control group that receives perhaps the standard of care, or a placebo, which is a substance that has no therapeutic effect. Researchers design phase 3 studies to demonstrate whether or not a drug candidate offers a treatment benefit to that specific population, always looking to provide more safety data, and then really serves as the basis for the product labeling, if you will. So when these trials are completed, the researchers look at the results and decide whether the drug has demonstrated effectiveness and an acceptable safety profile in treating that disease. And if so, they submit an application for approval. The FDA then considers, the application for marketing authorization. And sometimes these trials are called pivotal trials. And I also want to emphasize, sometimes it's overlooked, but there are phase 4 trials. So even after medicines are approved for use, there are still ongoing long-term clinical studies designed to even better understand the risks and potential benefits of that approved medicine over time. These clinical trials often assess how the drugs are working in the real-world setting and can evaluate thousands of patients over many years. And this information, as is the information collected in the other phase of the trial, are really important to payers, health insurers, and other bodies that make decisions on coverage. So again, this is a pretty traditional way to think about drug development. Over the years, sponsors have partnered with the FDA and other regulatory agencies to innovate in how trials are conducted. And this has led to finding methods where appropriate, to generating more information in each study that can lead to a better understanding of the benefits and risks of drug candidates more quickly, and even to combining different phases of drug development. And so the result is sometimes cutting the time significantly to filing for approval, or just as importantly, to stopping a program so that patients are not exposed unnecessarily to an agent that won't provide sufficient benefit or has too much risk. So the FDA has developed opportunities to facilitate and expedite development and review of new drugs to address unmet medical needs in treating serious and life-threatening conditions. And so, these programs are intended to help ensure that therapies for serious conditions are approved and available to patients as soon as it's concluded that these therapies' benefits justify the risks. And these expedited programs include things such as fast-track designation, breakthrough therapy designation, priority review designation, and accelerated approval. Today, I'm just going to focus on the accelerated approval pathway, and then I'm going to talk a little bit more about the Duchenne field in particular. So first of all, what does FDA approval mean? Well, it means that the drug, the data on the drug's effects have been reviewed by the agency, and the drug is determined to provide benefits that outweigh its known risks for the intended population. And the approval process takes place within a very structured framework, and I won't go into all the detail around that framework, but it's really around, broadly speaking, the analysis of that condition and available treatments, the benefit and risk information that's been derived from the clinical trials, and then the strategies that are employed to manage risks, the risk management plan and risk management and mitigation strategies, which is very important in drug development. And so in some cases, the approval of a new drug is expedited. So accelerated approval can be applied and has been applied to promising therapies that treat a serious or life-threatening condition and can provide therapeutic benefit where treatments may not exist, or benefit over available therapies. So this approach allows for approval of a drug that demonstrates an effect on a quote unquote surrogate endpoint that's reasonably likely to predict clinical benefit, or on a clinical endpoint that occurs earlier but may not be as robust as the standard endpoint used for approval. And so this accelerated approval pathway is especially useful when the drug is meant to treat a disease whose course is very long and an extended period of time is needed to measure its effect. And so instead of doing a study that takes 7 or 8 years, this pathway allows more proximal measures of disease progression to be analyzed. And this pathway, the accelerated approval pathway, was established in 1992, and many drugs have been successfully developed using this in HIV, in cancer, and then very recently, as you know, in Duchenne muscular dystrophy with exon skipping agents, and very recently, a drug for Alzheimer's disease. After a drug has received approval through this pathway, drug companies must conduct confirmatory studies. And these are studies that confirm how well a new treatment works after it was shown in early phase clinical trials. What the FDA needs to ensure is that these confirmatory studies are conducted and completed in a time frame that was agreed upon to confirm the benefit. And just touching on Duchenne, I'm sure everyone is aware that there have been 4 exon skipping drugs approved by the FDA via the accelerated approval pathway using dystrophin as a surrogate endpoint. The FDA has put out a guidance for what they call dystrophinopathies, and so that does include Duchenne. And this pathway has allowed these exon skipping drugs to be approved while confirmatory studies are ongoing to confirm their benefit. And again, this is a great example of where the FDA has really attempted to understand the severity of the disease, the morbidity and mortality associated with the disease, to try and get therapies on the market sooner while these confirmatory studies are moving along. And we also recognize that just over the last several years, there have probably been over 25 drugs studied that in their trials did not show either efficacy or on their pre-specified primary endpoint or acceptable safety. In most cases, the trials led to completely stopping the advancement of those candidates. And in a few cases, the sponsors reevaluated what went wrong and looked carefully at their study designs and asking questions: did we study the right patient populations? Did we get the dose and dose regimen right? Were the right outcomes used? And I think that plays into some of the discussion that we're going to have here with the DVA with Christine. And so it's really important to be thinking about the study design itself and how a drug is being evaluated, and if the right tools are being used to truly understand that treatment effect. And in all cases, sponsors are hopefully learning from each study, and I know that there's recognition of the sacrifice, burden, and hope from the patient and physician communities to have more choices on the market. I'll stop there and hand it over to, I'm happy to take any questions, or I can hand it over to Brian.
Thanks, Ash. I think we're saving questions for the end, if that's okay.
Okay.
And pleasure to be here with you all. Thanks for the invitation, Christine, and to be on a panel with Pat, Ryan, and Ash. Very grateful for this moment. I'm going to add a little more color to some of the things that Ash just shared. There's some very important historical perspectives that I can't help but sing the praises of the FDA and the theme of public safety. And so I'd like to give a historical perspective on that, but meanwhile we're waiting for this slide to come up. Here we go. Okay. So before I get into that, I just want to put a nod out there for all of you out there that the care considerations that are out there, whether you're in a trial, been in a trial, thinking about a trial, there's nothing about your care that should be sacrificed. So I really want to underscore this point, that never forget that there are very, very, very wonderful care guidances that have been established, a lot of it driven with PPMD's heart and soul behind it, to make sure that those are things you would never forego, having anything to do with being in or out of a trial. And I also have to put a nod out there for our moms, that there is evidence that carrier moms, there are potential risks that can be very much well managed by medicine. So our moms need to make sure their hearts check out well. And so I have to put that nod out first. And now let me dive into the talk. Next slide. So the FDA, as Ash had mentioned, comes from a very, very important background. And for those of you that kind of bash the FDA, which I've heard, there's actually a lot of history that you have to appreciate, and it's one that I deeply appreciate. So the history of the FDA really is built off of tombstones. The truth is, most of them were children. So the three pillars, I have examples of really the horrible tragedies that led to the laws that were enacted by Congress. So who is the boss of the FDA? It's Congress. FDA answers to the CFRs, the congressional legislation that directs them of what they are to do in the theme of public safety. And as Ash mentioned, back in 1900, this is 120 years ago. If you imagine prior to 1900, there was the Wild West out there. Anyone could sell anything and say it did whatever they wanted to say it could do. And this Biologics Control Act was triggered by 13 children dying in St. Louis after receiving a diphtheria antitoxin, which unfortunately was contaminated with tetanus. So that empowered the FDA, got them started to make sure that companies that sold things had a label on what was in it. So that was a good start. But as Ash said, that wasn't good enough. Next tragedy, the elixir sulfanilamide, 1937. This is the next generation penicillin, which hailed as a miracle antibiotic. And it was by a clever team. They dissolved it in a solvent called ethylene glycol, which unfortunately is your antifreeze in your car. Had a beautiful color, smells good, tastes good, but it is absolutely damaging to the kidneys. And there were over 100 people that died from the company that marketed this from Tennessee. And most of the individuals that died were children, again. So that led to laws that now, with the Food and Drug Cosmetic Act, 1938, led to must have things that are proven to be safe. These are things I think, if you think about the pillars of the FDA, so important. And then in 1961, which really isn't that long ago for some of us, the idea that there was a very important drug being marketed that was the first safe sleeping pill, especially for pregnant women. Thalidomide turned out to be highly teratogenic, and you see this picture on the bottom right of children that were born with exposed to thalidomide, which led to this condition called phocomelia, which you have these truncated, shortened limbs. And moreover, on top of that, there have been estimates there are probably 20,000 babies at the time that were born from this condition, and there were probably up to 80,000 miscarriages. We had a pharmacologist at the FDA who blocked the marketing of this drug in the United States, despite it being marketed heavily in the UK and Germany. And this further enforced the third pillar of the FDA: label, safety, and efficacy. And that's the foundation that I think we all have to appreciate and thank our FDA colleagues. Next slide, please. So here's a slide Mindy Leffler shared with me that I think says a lot of things. You would say, and as Ash had mentioned, it's somewhat deflating that of all these efforts in DMD, we still don't have a full approved DMD therapy. But I'd like to just put a nod out to Pat in PPMD. None of these would have happened. None of these existed before PPMD started. If you think about it, these are all things that have really moved forward because of the research that has been brought to bear in DMD, which to me, that is a great step forward. That is a huge nod to all the people that sponsored this work, the scientists, the sites, all the families, all those men, all those boys, all those families that contributed to this body of knowledge. It is tremendous. It has moved our field forward, and can't thank everyone enough. This is a community effort, and although they have not met the goals of where we want to be, it has helped move us such that we are in such a better position now than ever. Two other things to take from this is what I would consider really the lead up to what Sasha is going to talk about and what Christine is going to talk about. Is so far the interventions perhaps have not been efficacious enough. The mechanism of actions made a lot of scientific sense, but they couldn't deliver in the clinic. And the other thing that is also lurking out there is maybe the endpoints that have been used have been insensitive and have not been able to pick up important changes that matter to boys, men, and families, yet statistically didn't quite hit the thresholds. So let me hand it over on the next slide to Sasha, please.
Thank you, Brian. I think it's fair to say that we all want the same thing. As a pediatric pulmonologist, as well as just fundamentally as a parent, I want to see a treatment for DMD that works, that provides a clear benefit for boys and young men with this disease. What we don't want are claims that something works when it really doesn't. And we certainly don't want to see a treatment stopped, a trial ended, thinking that it did not work when it really does, but our endpoints were just not sensitive enough to detect or measure that benefit. In order to achieve this goal, we really need to have optimal endpoints that reflect a deep understanding of the natural history and how endpoints may change across the full spectrum of DMD, from young boys early in their disease progression, to tweens edging closer to loss of ambulation, to older teens transitioning to wheelchairs, and to young men navigating therapies for worsening cardiopulmonary function and loss of independence in their activities of daily living. Endpoints must be reproducible, not just in one country or one site, but across many sites in many countries around the globe. We have to understand how maturation affects those endpoints, as well as how standard of care therapies such as corticosteroids affect them. We have to understand which endpoints are truly objective and which are subject to motivational bias. We have to understand which endpoints are clinically meaningful and how much change in those endpoints is needed to reflect a clinically important change. Words and phrases matter when we talk about efficacy and whether outcomes are significant, when we talk about minimal clinically important difference and how that differs from minimal detectable change. We know endpoints differ in their usefulness across the DMD spectrum and depending on the mechanism of action of the intervention being studied. And several examples are shown here on this slide, and I will go through those that are highlighted in magenta in a little bit more detail on next slides. Many of the endpoints on this slide will look familiar to those of you who have participated in clinical studies. The muscle biopsy, the 6-minute walk test, the North Star ambulatory assessment. These are all well known in the DMD clinical research community. What may not be as well known are endpoints that measure cardiac function via MRI, circumferential strain, late gadolinium enhancement, as well as pulmonary function, a subject near and dear to my heart as a pulmonologist, percent predicted forced vital capacity, and peak expiratory flow. In addition, newer endpoints strive to reduce clinical trial assessment burden by being able to be done in the home, remotely, in a decentralized fashion. These are the 95th percentile stride velocity and the Casimir digital video assessment, or DVA. And finally, patient and caregiver reported outcomes, or PROs, are a very important part of the equation to show that a treatment or intervention reduces the burden on boys and men with DMD by improving their quality of life. A new DMD-specific PRO has been developed at the Center for Health and Technology, or CHET, that hopefully will be an improvement over other PROs that are not disease-specific. This was presented at the MDA conference earlier this winter and was very exciting to see. All of these endpoints are important depending on the stage of disease progression at the time of the intervention. And today, Christine will give you an overview of the DVA and how that potentially adds value and increased sensitivity compared to other clinic-based functional assessments. To understand the meaning of an outcome measure or biomarker, one has to understand the context, in this case, the natural progression of the biomarker. And I wanted to call out a serum biomarker, creatine kinase, which is a marker of muscle damage, as it illustrates several key points. This slide shows a figure from a paper that was published in 1986, but still holds today. They looked at creatine kinase natural history in boys and men with Duchenne. Just to orient you to this graph, on the y-axis, or the vertical axis, is the blood plasma level of creatine kinase. And this is plotted against age in years on the x-axis, which is the horizontal axis. As you can see in this natural history data, CK decreases with age in the absence of treatment. This is just observational. This decrease does not represent an improvement, but rather reflects the underlying fibrofatty replacement of the muscle tissue. If one were to see a decrease in CK in a 10-year-old from, say, 10,000 to 7,000 following treatment, it would be important to consider the natural history, which could explain that variability. If, however, one were to see a decrease in CK in a 6-year-old from 30,000 to less than 300, which is the normal range, in a short time frame following a treatment, that would make a stronger case, although the mechanism of action of the treatment would have to support that. This trend would also hold true for Becker, although the X and Y axes would be different scales. As this audience knows all too well, DMD is a progressive disease, starting early in life with inflammation, as seen in the leftmost panel in a biopsy from a 4-year-old, progressing over time with increasing amounts of fatty replacement and scarring, which you can see in the rightmost panel in a biopsy from an older adolescent. Becker's is also progressive, but again, over a longer time scale. Once fat and scar replace muscle, the changes are essentially irreversible. There is likely to be a tipping point at which the dystrophic process has progressed too far for certain therapies or interventions to achieve clinical benefit. Depending on the mechanism of action of a therapy, there may be an optimal time to intervene to achieve maximum clinical benefit. Although treating at a later stage may still provide meaningful clinical benefit if it could stabilize or slow down the process of fatty replacement and scarring. Muscle biopsies are the gold standard to demonstrate treatment effect. And while they are invasive, they remain critical as they provide direct evidence of the mechanism of action and treatment effect. As an example, if the mechanism of action of a treatment is anti-inflammatory, it would need to show a decrease in inflammation at the tissue level to be compelling. If the mechanism of action is anti-fibrotic, it would need to show a decrease in fibrosis in post-treatment muscle biopsies. If the mechanism of action is to restore dystrophin, it needs to show dystrophin. Whatever the mechanism is, you have to be able to show it at a microscopic level. Imaging is a non-invasive biomarker that holds a lot of promise, and a lot of work is being done to make it an FDA qualified endpoint for clinical trials, as we heard from Dr. Vandenborn on Wednesday evening in her chat with Pat Furlong. Will it ever replace the muscle biopsy? This is a really important question, and it remains to be seen whether it can indeed provide adequate proof of that mechanism of action, as well as truly differentiate between healthy muscle, inflamed muscle, and dystrophic or scarred muscle. At this juncture, the jury is still out on this, but it's definitely something to watch closely. Similarly, work is being done in cardiac MRI to understand the natural history and how it might help in clinical trials. Left ventricular ejection fraction, or LVEF, is a measure of how efficiently the heart squeezes blood out of the body and can be measured in two ways, one by echocardiogram or by cardiac MRI. You can see in the panel on the left a publication from Dr. Khan Hoare that there is variability, but a clear decreasing trend in LVEF as measured by cardiac MRI as children get older. This seems to be more sensitive than LVEF as measured by echocardiogram, meaning that it shows changes earlier than can be detected by echo. This is important in terms of clinical management so that medications can be adjusted properly and in a timely fashion, but also in clinical trials, as it might help provide evidence of a treatment effect on the heart muscle. Pulmonary function testing is also an important biomarker, particularly in older children with DMD. And you can see in the right panel a figure from Dr. Hank Mayer's publication that forced vital capacity and peak expiratory flow, two key pulmonary function test measures, increase with age and then decline in boys and young men with DMD. If you look at the percent predicted of both of those measures, which normalizes the raw value based on height, gender, and race and ethnicity, there is a linear decline with age that starts at around age 6 to 8. This is a test that is non-invasive and can be taught to young children. I've done this myself, making it an attractive endpoint for clinical trials, particularly when we understand the natural history trends. Understanding natural history is incredibly important when trying to interpret outcome data in clinical trials. And thanks to all of you who have participated in observational studies and placebo arms of clinical trials in the past, there are robust data sources in DMD in terms of real-world data. There are 27 different data sets across many countries and regions of the globe involving thousands of patients. You can see that a few listed here: the Collective Trajectories Analysis Project, or CTAP, the Cooperative International Neuromuscular Research Group, or Synergy, and the Critical Path Institute, or the Duchenne Regulatory Science Consortium, or DRISC. And you can see there are thousands of patients, and you can see the countries listed as well. These are great resources for helping design clinical trials, but one of the big questions is whether all that data that has already been collected could be enough to serve as a comparator in a confirmatory trial. And many are trying to make this case to the FDA. This is one step towards that goal. This is a publication, which is the end result of a fruitful collaboration between academic institutions and CTAP. This study demonstrated that real-world data was comparable to placebo data for the 6-minute walk test. Just think about that for a second. Real-world data is comparable to placebo data. This group plans to do the same analysis for the NSAA. Very exciting work, to be sure. And speaking of the NSAA, a talk on endpoints in DMD would be remiss if it did not mention the NSAA. And here you can see what I am sure is a familiar figure from Dr. Mentoni, something that we have taken to calling the Hay Bale Plot. It shows the wide variability in NSAA scores on the y-axis, plotted against age and years on the x-axis. And there is a ceiling effect that the maximum score is 34 points. And most non-DMD children reach this ceiling by about age 4 to 5 and stay there. And while you can see that boys with DMD are below that ceiling. Again, natural history matters in order to set clear expectations. The goal, depending on when the treatment is initiated, is to improve, stabilize, or slow the decline in order to change the trajectory of the disease. All of these outcomes would be considered home runs for each stage of DMD. So disease stage at the time of treatment or intervention matters greatly in terms of expectation for what that treatment can do. This has to be considered by all of us, physicians, sponsors, regulators, and even families when it comes to evaluating study design, setting expectations for what treatment benefit looks like, as well as in the interpretation of clinical trial results. So in summary, Brian and I hope we've been able to give you helpful information about the importance of following the standard of care, the importance of safety, and how it has been and will continue to be a key pillar of the FDA, how each boy and man with DMD has his own trajectory, which may be more pertinent than the endpoint itself, and that real-world data exists across many and multiple sources. And our challenge as clinical drug developers working in this space is to prove in a robust clinical trial that a treatment is safe and efficacious for boys and men with DMD with an appropriate sample size that balances benefits and risks. And we as a community must leverage the real-world data in designing our studies, assessing endpoints, and in considering real-world data for use as a comparator so that we can minimize the number of boys and men in placebo arms if that's possible. And I just want to thank Christine and Mindy, as well as Ryan and Pat, for the opportunity to speak here today. We'd also like to thank Ash for his thoughtful comments. And we all look forward to taking questions at the end. And with that, I'll hand it over to Christine to talk more about Casimir.
Thank you, Sasha. And Stephanie, I'm going to have you bring my slides back up because I can't be trusted to do them myself. And you notice my location has changed, and landscapers, every room I go into, the landscapers seem to follow. So let me know if you have trouble hearing me, and I will put myself in a closet, I guess.
Christine, can I make one quick comment?
Yeah.
Just on the real world data? I think that, you know, everything Sasha said is so critical for all of the reasons. I would also just add, in terms of real world data, the Duchenne registry has been absolutely an amazing resource that looks at real world data from a number of different sources, from electronic health records to clinical trials, and then all of the patient reported outcomes that have come in. So, and then trying to tie those patient records to clinical records, patient reported outcomes to clinical records. It's been a tremendous undertaking by PPMD. So I just wanted to put it out there.
Thank you, Ash. Thank you, Brian, and thank you, Sasha, for everything that you added to the conversation. Super educational for our community to understand history and kind of what the challenges are today, and then the progress that we're making today. So if you can go to the next slide, Stephanie. So I'd love to start with this slide because this is how I look at our Duchenne community, right? That each patient should be considered as their own N. And not everybody should be asked to be able to climb the same tree given where they are. And that's what Sasha was just talking about, is we really need to understand the trajectory of their disease when we put them into clinical trials, when we put our participants into clinical trials. Patients may be in that age of actually gaining some milestones at 5, 6, 7, 8, or that same patient population may be losing milestones and be on a downward or decline trajectory. And we put them into the same clinical trial in that same cohort of patients. And we ask them all to do one test that's going to evaluate whether or not the intervention is beneficial. We're going to get varying results. And so this is one of those slides that I always like to start with so that we can kind of like set the tone of what we're going to talk about today. In the next slide. I'm keeping an eye on the time. And so for Mindy and I, and you guys, know a lot about our story in the past. We were, we were part of the Ejection review, and we were part of the Ejection safety study. And it was Mindy and I that took our boys home, gave them infusions once a week, and then began to notice things that were happening outside the clinic. Things were different. You've heard the stories before about Jet had stopped snoring, and it wasn't me that noticed, but it was his roommate. Aiden was able to get in and out of our SUV much easier, and we'll see those videos in a little bit. And it became really clear to us that there were things happening outside the clinic that were truly meaningful. For Jet to stop his snoring, it meant that he didn't have to have nighttime ventilation. For Aiden to be able to get in and out of the car without any help, well, that meant that he wasn't embarrassed in front of his middle school when his mom had to pick him up and put him in and pick him up and take him out. So meaningful benefits. And along that course, we began to realize that the concept of interest that we were working with was something called ease of movement. Not only was Jet able to breathe better at night, but he was able to reach over and pat his dog from his power chair and bring his arm back up to the power chair without asking for help. And so what we realized is that what is Duchenne? Well, Duchenne is a neuromuscular disease that becomes difficult to move. It's just simply hard to move because you're losing muscle mass. And the next slide. And so what do we watch? And so all the parents that are listening to me right now know exactly what I'm talking about. And so what are the activities that you watch when you're trying to understand if your loved one who's affected by Duchenne is progressing in their disease? And what's the one thing that you turn away from when you just don't want to see it that day? And I know you understand what I'm talking about. So when Jet was 6 or 7 years old, it was how he was getting into our house. Could he take those 3 or 4 steps? Or did I have to start thinking about putting a ramp in? As he gets older, is it the way that he eats? And is he not going to want to go out to eat anymore with his siblings because he's dropping food on his chest and he's embarrassed about it? And as boys get older, is it the ability to reach for that joystick and actually get themselves around their homes by themselves in their power chairs? And so you all know that those are the things that you look at when you're making decisions in your home about how to take care of that person. And so Mindy and I took that concept and we put it into something that's quantifiable called the Duchenne Video Assessment. And does that activity you watch change over time as they progress? So as Jet became from ambulatory to non-ambulatory, I would look at new things, different things. But there was always something that I would be looking at. And what was the loss or the gain of a milestone that happened? And so for us, the gain of a milestone that happened was he was able to breathe at night without snoring. That's a milestone. He was able to pull his arm back up onto his power chair. And then healthcare providers, our PTs and our awesome clinicians that are in the audience, what is it that you watch when your patient comes into clinic? Do you watch the way that they get up from a chair or off the examining table? Do you watch the way in which somebody adjusts their eyeglasses during the visit? Do you watch the way in which maybe a young man crawls his arm up and you can actually predict, hey, he's probably losing the ability to do that, or geez, that changed from the last exam. And the next slide. And then how do these observations affect daily life? Like I said, as your child loses those milestones, that affects your childcare at home. It affects work schedules, equipment considerations, transportation. It affects where you live, the selection of school, and the interior layout of your home. Think about it when you go to clinic and they ask you to walk down the hallway. Your PT is watching not only how fast they walk down the hallway, but that PT is actually watching the way in which they walk down the hallway. What is the lordosis on their back? How high are their heels up? Are they on their toes a little bit more? Do they have to kind of maneuver their hips and their shoulders so that they don't fall over? Do they actually walk a little bit faster because they're afraid that they're on their toes and they might fall before they get to the door, if that's the point that they have to stop? And so taking all those things into consideration, again, we're looking at the way in which people move. If I wasn't feeling good today and I had a sprained ankle and I came into this room, this office room to do the webinar today, I'll get here. Like, I'll show up, but it just might not be so pretty because I'm going to walk a little differently. Well, we notice the same thing happens with boys with Duchenne. In their disease, they progress. They will add compensatory strategies to be as independent as they can until they can no longer do it anymore. And parents and caregivers, you know exactly what I'm talking about. So should we consider capturing them as important indicators of progression, stabilization, or improvement at home? And how can we do that? And we all know that FDA, as we've talked a lot about today, they approve drugs on how people feel, function, and survive. And feel, function, and survive includes in their home environment, where they have to navigate their own bedroom, kitchen, living areas, and restrooms. And the next slide. And so, as I said before, so we can almost skip this, but Exondys 51 is where Mindy and I first started noticing that there were changes happening to our kids We thought that those changes were so meaningful to daily life. We concluded that with experts in the field, PTs, clinicians, KOLs, our advocacy groups, including PPMD, and everybody said, yeah, actually, those are important and they should not be missed. And so is there a way in which we can capture them? And the next slide. And so this is just quickly, I think if you can play this, Stephanie, that would be great while I talk. Most people have seen this slide before in these videos. This is what Mindy identified as something meaningful to her and her family. If Aiden was no longer able to get in and out of the car, it meant that they had to get a different car that he could get in and out by himself. And so she said, if this treatment's going to work, I'm going to see it here, because this is the thing that she was watching as a parent. Perfect. And as you can see at the beginning, Aiden is frustrated. Whoops, he puts his arms up. He can't get into the car. And the smile on his face tells it all at the third time point, which is about 7 months into treatment. And the next slide. And so what is Casimir? So Casimir was started by these two moms. We are a fully remote organization that utilizes technology to capture prescribed assessments in the home in a non-invasive manner. And so we know that DMD is a progressive muscle disease. It becomes hard to move. So we want to see how patients are moving in their home. What does it take for them to eat? Are our patients going down here to eat, or are they sitting up here to eat? And does that change over time given an intervention? What are the compensations that a patient will use to complete that activity? So we have prescribed activities that we've created with the help of all of you, KOLs and our caregivers, and our participants, to tell us what was important to you. And then we have 2 additional videos that we like to capture in home. And one is called New Ability, and the second one is Caregiver Choice. New Ability is really important because the day that I walked into clinic and I told the nurses that Jet was able to smile again, they went, that's great. Oh, we can see it. But they had no place to put it. And yet it was such an important milestone that he gained back. So New Ability is really important. Caregiver Choice is really important because that's that one thing that you're watching. You're watching that child at home or that young man at home. It's probably the first thing that you'll pick up on that changes. Is he able to reach in his pocket and pull out his wallet? And he hasn't been able to do that in years. And why is that meaningful? Because maybe now he can take the T into his job in Boston and he doesn't have to rely on help. So really small things, really nuanced improvements are improvements of a lifetime for these young boys. And I don't want to miss them. Caregivers, you know what we're looking at when we evaluate kids, when you evaluate your sons. Are they walking their hands along the floor to stand up? Are they swinging that leg over the riser to get up? Are they crawling one hand up while carrying the other one to be able to get to their upper body? Those are really important milestones that we either adapt or we shed due to an intervention. And if we shed them, we need to capture it because it's important. It means more independence for these boys. And so we have created a methodology in which we can quantify these compensations into a severity score. And if that severity score decreases in a clinical trial, that means that child is gaining those acquisitions back. And the next slide. Why do we do remote assessments? Because as parents, it's really hard to travel with our young men with Duchenne. I think we all know that. It decreases patient burden. And I think we end up with a better trial experience as a result of it. If we can capture some things at home and we don't have to put our kids on airplanes, if we don't have to do a 13-hour car ride to get to the clinical site, that's great for us. We don't have to spend the money. We don't have to get childcare for our kids, other kids. And we don't have a child who ends up in clinic at 9 o'clock in the morning when he's not even used to getting up till 9 o'clock in the morning, being assessed by a strange assessor. He's being assessed in his own home where he's comfortable by mom, dad, or another caregiver who he feels comfortable with. We can capture and we can demonstrate change over the short time. And we have done this in several different trials in only 12 weeks. And so other outcome measures may take a lot longer, but we begin to see changes in about 12 weeks. We believe it better reflects function in daily life. We don't have the confounding factors that I talked about: travel, fatigue, clinic fatigue, medical anxiety, and trauma and depression in vacation mode. We don't arrive at a new city and take our kids all around the city just to see it, to make the trial experience better because we feel guilty that we're putting them into the clinical trial to begin with. And I know moms know exactly what I'm talking about, and dads. In the next slide. The other point I want to make on this slide too is that we're able to open clinical trials up to participants who may not be able to participate. So there's lots of people who aren't able to travel every week, every 2 weeks, every month, every 3 months. If we can replace some of those clinic assessments with a remote assessment, we actually improve and increase patient compliance as well as the ability to participate. So why do we do these? Because they actually tell us the story of what's going on. They tell us the story of what's happening in the home with our participant. It's not just a snapshot in time, like you show up in clinic at month 3 and this is what happens, and this is what we're assessing in 1 hour. We actually get to see over a period of 12, 24, 36 months what's happening to this person who's in the clinical trial. It's an important part of the information gathering process, especially to our study designs. If we can implement something like a remote assessment early, it can tell us what we need to put in our P3 and be a little bit more predictive and on target. It also can tell us what trajectory the patient is on prior to going into a clinical trial. As I said before, we want to know if that patient is in the decline phase, stabilization phase. It's really important to inform us. And we can go on to the next slide. We are coming up on the hour. So how does it work? Patients and caregivers are asked a series of questions. We put patients into different cohorts. We don't ask them to do tasks that they can no longer do because of their disease. The video capture is usually about 4 to 5 to each patient cohort: early ambulatory, late ambulatory, early non-ambulatory, and late non-ambulatory. And the whole process takes about 15 minutes. Your video is uploaded, stored in a HIPAA GDPR compliant cloud, and then blinded raters who are trained by Casimir come in and they score the videos according to the data management plan. It's important to note that the PTs that are scoring these videos are blinded to the time, and you often have 3 PTs who score it. If there's a discerning score, then the majority wins. So if there's disagreement, we have a way to adjudicate that. In the next slide. This is just a little bit of what the screen looks like when it's in the app. Again, those videos are not locally stored. Everything is reviewed by quality and compliance. Reshoots are optional by the sponsor. Training is provided to the caregivers. And then the videos are scored by central raters. And we can go to the next slide. Just really quickly, we can play this video, time point 1. I want you to watch how this child gets up off the floor. And time point 2. And I think we would all say that time point 2, we see that young man, he's got a wider base of support. He's crawling his hands back, and his scorecard would reflect that, which is on the next slide. He's getting a little worse in time point 2. If we were to see some of these walking along the floor or hands not on thighs, we would score his scorecard appropriately, and it would mean that he would be shedding at time point 2 some of these compensations, which with our physical therapists and KOLs in the field would say he's improving. And the next slide. And Ryan just yelled at me if I have to stop. And so we can skip this one too, because we've kind of already talked about this. We're going to meet each patient where they're at. We're going to quantify ease of movement, not speed of movement. We're going to capture those instinctive adaptations to healthy movement patterns. We're going to capture all nuanced and significant compensations, and we're going to allow for each patient to have their own compensatory strategy and provide a window into patient function in the home environment. And next slide. We want to actually ask the research question: is the intervention demonstrating meaningful change over the course of 3 months, 6 months, 12 months? Is there stability or decline? Can we show continued improvement longitudinally, past 12 months, 24 months, 36 months? Is there stability or decline? We can do this remotely, capturing these videos and having PTs score them. And then is the intervention showing continued improvement 5 to 15 years from now, really trying to get to that question about gene therapy and what it holds for our future. And the next one. This is just an example of some of the things that we capture using the home-based video capture system. And the next slide. This is the scoring scale development that we did in the testing. We now have a validated scorecards. And the next slide. I'm going, Pat, as fast as I can. And then where Casimir is today, we have validated task selection, validated capture procedures, validated scorecards. We've had a 7-patient source material study, a PT certification program. We are in the FDA qualification pathway. We are in discussions with the EMA. Reliability testing has been done, validity has been done, and a caregiver questionnaire has been given. And overwhelmingly, everybody said that they prefer home-based capture, which is important. In the next slide. What do we need? We need test and retest. We need longitudinal data. We need some natural history data, and we need power calculations for cohorts. The goal is for Casimir to become a primary or secondary endpoint that informs all stakeholders of the capabilities of a treatment. And the next slide. How are we going to get there? We actually need your help. So we need the help from everybody, all stakeholders. So whether it's industry, academia, our advocacy partners. We will be recruiting an ARISE study this summer. We will be recruiting up to 100 patients affected by Duchenne. We'll include a natural history cohort, which we talked about with Ash and Brian and Sasha, which is so important. All stages of Duchenne, I'm going to start with the 4 to 6-year-olds because there's some maturational differences there, and we'll be expanding quickly to include others. And the next slide. How you can help, email me, text me, send an email to info@casimirtrials.com. If you're interested in participating, we'll put you on the list. We'll be in touch with you shortly. This is going to take our community effort. We saw what happened last year with COVID. It was amazing when government, industry, patients, public all came together. We were able to get a vaccine and many vaccines in a record amount of time. Our boys don't have any more time. We have over 40 failed clinical studies right now. We may have a way to gain insight on what's happening with these kids. But if we don't all work together and become efficient, it's going to be longer, and we could have more failed studies. So it's important. If we can all do this together, it would be great. And I want to thank Pat and Ryan and PPMD, Ryan's Quest, Michael's Cause, Dine, and Sarepta, and Vertex. So far, you guys have been fantastic partners in helping us get this done. I hope I did it on time.
That was, no, I know we're a few minutes over, but time for a few questions, if you can. I know one came in here. Can you participate in the ARISE study if you are in the current Casimir study?
You know what? Just email me and let me figure it out. Yes, I believe you can.
Great. Great.
We're working closely with FDA to design this study, so there's still some outstanding questions with FDA.
Great. Yeah, and you talked a little bit about the qualification process. Can you describe that in a little clearer detail for families so when could we expect this to be a qualified endpoint that could go into a trial? We do know some trials are looking at it as what they call exploratory, but they're utilizing it, correct? So it could be supportive evidence. But could you talk a little bit about when the FDA says this is a good endpoint, secondary or a primary?
Yeah, so I'll give you a little hint on FDA qualification that I don't think we knew when we went into this, Ryan, and that's they look at this process as if you were developing a drug. So it's like asking, one of our sponsors, when do they anticipate approval? It's probably going to be a couple years before we can anticipate full qualification, but we are working with them in lockstep with them. It's not a drug, so we're not going to see any side effects. So I think right now they're waiting for longitudinal data. So this is our next best thing to do: test, retest on the instrument itself, and then longitudinal data. That's what's it's gonna take, and I think probably at least 24 months of longitudinal data, and then I would guess qualification 36, 48 months from now. Yeah.
And that process is frustrating, to say the least, for a lot of sponsors and academics to get things qualified. But so you also mentioned COVID. Quite frankly, the other thing about COVID is we've learned about the need for tools that you can do at home. Telemedicine has grown. So that's just another sort of reasoning around developing better drug development tools like this. We've been happy to partner with recruitment. We will continue to do so. We believe in the work you're doing. I don't know if any of the other panelists have thoughts or closing comments they want to make. If not, I'm going to hand it to Pat to close us out, if anyone on the panel wants to say anything.
Thanks, Christine. Thanks, Ash. Thanks, Sasha. Thanks, Brian. So one of my thoughts or questions to you, Christine, is, in this discussion, we're very familiar with the Center for Drugs. And so just wondering about, as you qualify this, are you talking with and working with both CBER and CDER?
Yeah, so a lot of our conversations have been with CDER specifically. Conversations will begin with CBER very shortly, but we mostly work with Critical Path, who is our liaison to both CDER and CBER and the COA group at FDA.
Yeah, so the Critical Path has been such a good partner for us with so many data sets inside and qualifying, as was Sasha mentioned, working on qualification of the MRIs, the biomarker as well. So these next years, we should have much more sensitive tools that decrease burden, and tools that really help families capture what's actually happening in the real world, rather than a single data point at a certain time, which is influenced by 1,000 million factors of mood, sugar level, willingness to participate, et cetera. So what you're doing is so needed by this community. We so appreciate it. And of course, we are thrilled that Vertex is here, Sasha and Ash, that you're with Dyne. So we see and hope for great progress in the future. Thank you for this Lunch and Learn. It has been really remarkable, and keep going. And to the degree that we can be of use, we want to be right there. So thank you all very much. And we'll see you again soon. Thank you. Thank you.
Thanks, everyone.
Thanks, everyone. Thanks,
Thank
Pat.
you.
Thanks, Ryan. Have a great weekend.
You too.
You too.